This is the audiobook version of Hacking Bigfoot, Deconstructing an Unknown Species by Marcus Daly, published in 2025. A technical glossary and endnotes with source material are available on the publisher's website, placement.pub. It's all about the hair. It has long been known by investigators that Bigfoot hair is very different from human or any known animal hair of any continent. Suspected orang pendek or Bigfoot hair is coarse with a blunt tip and no taper or medulla. A medulla is a spongy mass at the center of hair follicles. When examined under a microscope, suspected Bigfoot hair samples are consistently 65 microns wide. In 2001, investigator Adam Davies recovered suspected orang pendek hair samples on an expedition in Sumatra. The hairs were examined by the late doctor, Hans Brunner of Australia. Hans suspected the structural analysis to be either an undiscovered type of orangutan or something very closely related to an orangutan. Dr. Brunner used a classic microscope to compare the two sample hairs against all known primate hairs from that region that were available to him, such as orangutan, siamang, gibbon, chimpanzee, gorilla, sun bear, red-leaf monkey, pigtail macaque, tapir, and human. There was no match, so that led to the theory they belonged to an unknown species dwelling in Sumatra, presumably the orang pendek. Though Dr. Brunner was enthusiastic about the discovery, he did not run biomolecular analysis. Dr. Todd Disotell ran his own biomolecular analysis and was underwhelmed by Brunner's hypothesis. He suspected the samples were contaminated because they had human DNA sequences. In the 1990s, Dr. Henner Fehrenbach collected and studied many supposed Bigfoot hairs and determined that DNA testing will always return as either human or inconclusive. At first, Dr. Fehrenbach thought this was due to contamination. He deliberately contaminated dog and cat hair with human DNA and tested it, genetically confirming it as dog and cat hair. This demonstrated samples retain their species characteristics despite being contaminated. Everyone who has studied a suspected Bigfoot hair has concluded it doesn't yield DNA well. Hair is not living tissue and has limited value, especially for identifying a new species. Follicles with fat tissue are needed to acquire more data on physical characteristics from alleged Bigfoot hair. If there are no fat, fresh follicles, the hair will not be of genetic value. In 2009, a return expedition by Davies recovered more alleged orang pendek hair samples. This time, it was sent to Dr. Tom Gilbert, a genomics researcher specializing in ancient DNA and evolution. Dr. Tom was able to extract enough mitochondrial DNA to conclude that the hairs were either human or very closely related to humans. He was confident that the hair samples were not contaminated. An explanation of that can be found in the functionality of bioinformatics software. This technique is used to find regions of similarity between genetic sequences and fill in sequential gaps using the closest phylogenetic match it can find. If samples are too degraded, there are going to be numerous gaps. Those are considered to be unknowns. So, even if it was genuine Bigfoot DNA, the tests would show a human result, because that's the closest match for the sequence that can be bioinformatically determined. In no way is it a confirmation of Bigfoot being of Homo lineage or convincing evidence of anything.